this is the first question of passage 6. It says, how does the unit membrane model differ from the fluid mosaic model? So this passage is essentially kind of like a CARS passage in which you have two theories of membrane structures written below the unit membrane model and the fluid mosaic model. Well, so let's just take a look at A, B, C, and D. Let's just see which one kind of seems correct here. A says the location of proteins differs in the two models. So let's take a look at the unit membrane model. Where are the proteins in this model? It says a monomolecular layer of protein coats both the outside of the membrane and the inside of the bilayer, but the protein does not penetrate the bilayer. All right, so that means there is this layer of protein on the outside, but it doesn't go through the actual membrane itself. What does the fluid mosaic model say? It says there are proteins inserted into the bilayer. So this seems pretty correct, right? So the location of proteins does seem to be different in the two models. So let's take a look at the rest of the answer choices, but so far A does look like a very strong answer. It says B says the unit membrane model has a monomolecular layer of protein on each surface, while the fluid mosaic model has a bimolecular layer of protein on each surface. As we just stated, the fluid mosaic model doesn't suggest that there's a layer of proteins. It says the proteins are inserted within the bilayer. C says the unit membrane model has one layer of phospholipids, while the fluid mosaic model has two layers. Again, there is just no, well, actually, here it's a little bit different. We're looking at phospholipids, but in both cases, we're looking at a bilayer. So unit membrane, bilayer of phospholipids as well. And then for the fluid mosaic model, there's a bilayer, which means both suggest that there are two layers of the phospholipids. So C cannot be correct. And D says the unit membrane model contains dissolved proteins, whereas the fluid mosaic model is coated with a monomolecular layer of protein on each surface. Guys, like we've just been saying, the proteins in the fluid mosaic model are inserted into the bilayer, meaning D has to be incorrect, meaning A is the correct answer. All right, so this is question number 2 of that passage. It says, which of the following observations would invalidate the unit membrane model? Okay, so it says A, when a thin section of membrane is observed using a microscope at high magnification, 2 layers of phospholipid heads are observed. Well, this would make sense, guys. Remember, unit membrane model, it suggests that there is a bilayer of phospholipids, meaning that if you were to actually get a cross-sectional view, you would see those two, the bilayers. So right now what I'm drawing are the phospholipids. The circles are the heads, and the tails, well, the squiggly lines are the tails of the phospholipids. So because the unit membrane model does suggest a bilayer of phospholipids, that means you would see 2 layers of phospholipid heads, indicating A cannot be, A would actually fall in line with the unit membrane model or support the unit membrane model. In that case, it wouldn't invalidate the unit membrane model, meaning A has to be incorrect. B says thermodynamic measurements indicate that the phospholipid heads are exposed to water. The unit membrane model suggests that the polar hydrophilic heads would line up to interact with water, right? Yeah, so the tails point inwards and the hydrophilic heads line up on both outsides of the bilayer, so that these heads can interact with H2O. So this would actually fall in line with the unit membrane model. B would fall in line with the unit membrane model. So again, this would not invalidate the unit membrane model, meaning B has to be incorrect. When a membrane is frozen and then split from surface to surface, the proteins are observed within the hydrocarbon chains. The hydrocarbon chains are these, right? So the hydrocarbon chains are the hydrophobic tails that they're talking about. And remember, the proteins are not, they coat the membranes, but they do not penetrate, okay? They do not penetrate the bilayer. Meaning if you were to split up the layers and you do find proteins interacting with these hydrocarbon tails, well, that means that you know the proteins have to be embedded into the membrane bilayer in some capacity. Meaning C would invalidate the unit membrane model, meaning C is a pretty good answer choice, but let's check D. Thermodynamic measurements indicate that the phospholipid tails are hidden inside the membrane away from exposure to water. This is supported by the unit membrane model. It says that the tails are pointed inwards because they're hydrophobic and they do not want to interact with water. So D would actually support the unit membrane model, meaning C has to be the correct answer for this question. This is the third question of the passage. It says, if the small bumps seen when half of the membrane is peeled away were chemically shown to consist of the lipid cholesterol, how would the fluid mosaic model have to be modified? So just to reiterate this point, or to kind of go back to what this is referring to, let's take a look at this paragraph here. When the membranes are split along the middle of the lipid bilayer, so the top half, okay, so remember there's 2 layers, two phospholipid layers. They're going to peel off this top half, okay? They're peeling it off, okay? And then they're just going to get rid of this. And then they're going to look down on this layer, and they can see small bumps, all right? They assume that these small bumps are proteins. Well, this question is saying these small bumps were actually lipid cholesterol. So if these were actually lipid cholesterol, then how does the fluid mosaic model have to be changed? Well, A says the protein would have to be embedded less than halfway through the membrane. What they're saying is the proteins are maybe embedded over here or embedded like this. Well, that's not necessarily true. What could have happened is, let's say the protein was all the way, I should make the proteins orange. Let's say they go through all the way the membrane, but you peel off this membrane that has all of these proteins embedded in it, and now you're left with no proteins on this bottom layer here. So that means A doesn't necessarily have to be true. B says there could be no proteins in the membrane. Well, based on what I just suggested here, by the proteins being peeled off with this membrane, B doesn't necessarily have to be true either. The lipids would have to be embedded in the proteins. I'm not entirely even sure what that's talking about, but first, the lipids, it would have a very difficult time being embedded inside of the proteins, if the phospholipids especially, due to just the hydrophobicity of the lipids. And not only that, there would be no indication that the lipids would have to be embedded inside the proteins to make this theory true, where the proteins are actually embedded into the top layer where it's peeled off. So it leaves us with D. It says it would not necessarily have to be altered, but there would be less evidence supporting it. So yeah, if it were true that we peel this layer off, we look down, and it's actually cholesterol lipids as opposed to proteins, well, that doesn't necessarily disprove the fact that proteins can be embedded, suggested by the fact that these proteins can actually be in the top layer, but we would have to look for other evidence in order to support our original, well, at least the fluid mosaic model's original stance that the proteins can be embedded in the lipid bilayer. This makes D the correct answer for our question. All right, let's take a look at the last question of this passage. It says, which of the following is a weakness in the unit membrane model? The unit membrane model does not define the cell size. The cell size actually doesn't really depend. So remember, these theories are trying to describe the membrane structure. Neither the unit membrane model nor the fluid mosaic model describe how this would actually determine the cell size. So it's not necessarily a weakness that they don't talk about it. That's just outside of the scope of what they're trying to describe here. So A is not the correct answer. B says the unit membrane model does not suggest how the exchange of nutrients and wastes occurs. This is probably a weakness, right? So you have this model where you have a lipid bilayer, and you have these proteins. Let's draw them as squares, okay? Okay, these proteins are on both ends, both sides, and this doesn't necessarily tell you then how do nutrients and wastes cross the proteins and then cross the bilayers as well. So the fluid mosaic model tells us, hey, this can actually happen because let's say you have these phospholipid coming together again, and let's say you have some kind of molecule that needs to get through. Well, this protein, let's say some kind of protein like a tube, can actually help that molecule, that nutrient or that waste to get in or get out of the cell. But if you have something where the proteins can't even extend throughout the lipid bilayer, let's say you have a polar nutrient or a polar waste, it's going to have a very difficult time, even if it can cross through the proteins. How is that polar nutrient or that polar waste going to get through these hydrocarbon tails? Whereas in the fluid mosaic model, well, these proteins help those polar molecules get through, usually because the inside of these tubes or these tube-like proteins, they're polar, to allow the polar proteins or the polar molecules, to get through the lipid bilayer. So B seems like a pretty good answer. It doesn't suggest how nutrients and wastes would go through the bilayer, just as, whereas the fluid mosaic model does suggest a way for this to occur. But let's take a look at C. The unit membrane model does not act as barrier between the contents of the cell and the cell's external environment. That's not necessarily true. Even if this model was the correct model, this would protect the cell, the inner contents of the cell, from the outer contents of the cell. So C is not necessarily a correct answer or a weakness of the unit membrane model. And finally, the D says the hydrocarbon bilayer allows passage of polar molecules through the membrane. As I've alluded to before, the hydrocarbon bilayer, so these hydrocarbon tails, let's say I have this bilayer right here, you can't have a polar molecule get through, right? These polar molecules, plus charged or minus charged, or maybe they're hydrophilic in another sense, maybe they like to bond onto water, they can't, it's going to have a very difficult time going through this passage of hydrocarbon tails because, well, the hydrocarbon tails are hydrophobic and the polar molecule is polar, it's hydrophilic. So D is not a weakness because the hydrocarbon bilayer will not allow polar molecules through the membrane in the first place. This statement is just false. So that leaves us with B as the correct answer for this question.